Hi, boys and girls. Another episode of Rational Empiricist Rants. Today,
we got to hear some rants.
And I'm 25,900 today. It's my birthday. Everybody better wish me happy birthday.
Yeah, what'd you get me?
Half a cup of tea.
That ain't how that works. You're
supposed to get party favors. We came to the party.
Oh, well, no, you don't get no favors. I don't do that.
Ah, dang it.
Anyway, hi, everybody. Woolly Ignorant. And
hello.
Scott Duke's in the chat.
Yeah, he's just always around.
Secular Pagan Mom, Gur Streams. Oh, my God. Pixie Storms, Gur Streams.
I call them Pixie Storms.
Kink Streams now.
Saying about time. Well, guess what, Gur? We don't go by your time zone standards.
Yeah.
Your schedule.
Yeah.
More like non-standards.
Yeah.
Damn atheists and their lack of standards.
Oh, my God. I'll tell you, if anybody needs a godly foundation for their morality, it's Gur. I don't know. Anyway. So, Andrew, what you got for us today?
All right, so I guess should I share? Oh, hope I'm not roboting.
No, we'd like you to share.
Oh, okay.
That's
because you mute for a sec. Oh, I guess it's not doing that anymore because it used to. I used to robot when everyone else in the room was muted.
Oh, let's see.
Testing. Am I roboting?
Nope. It only happens.
Yeah.
It's sort of a StreamYard glitch, that every now and then that happens. And StreamYard also when you hear the echo when you first start talking, it takes a while for echo cancellation to kick in.
Huh.
So I don't know. Anyway.
All right. Let's see.
It's a fun technical talk.
Let me share this PDF.
There you go.
All right. Can I make it larger?
Well, if you made it skinnier, we could also make you full screen.
All right. Is this readable at all?
Very readable.
Okay.
I can read it.
All right. So I just created a short little thing here because I had some inspiration today after, going through some of SFT's older videos and just thinking about, what am I going to focus on today, basically. And so I kind of went back to this idea of phylogenetic incongruencies and the solutions we have to them now. And while I was doing another activity, I basically had this big inspiration and came up with this, or what I at least think is a useful analogy for what you hear about phylogenetic incongruencies often by creationists when they do actually address phylogenetic data. This analogy that I came up with basically for why this isn't actually a problem for common ancestry, but I'll eventually get to that. For now, I'm going to give a short little primer on some of the main phylogenetic incongruencies or the sources of those incongruities that are biological in nature. So we know these happen. We know that they're part of today's population genetics, for example. But anyways, So the central points from phylogenetic incongruencies in general, first of all, a phylogenetic analysis is basically, it's relying on comparison of, kind of using the word loosely here, but unique evolutionary changes. So we've already talked about, for example, that genetic similarities aren't really that crucial to phylogenetic analyses. It's more about seeing, okay, how is this state or character different from this other state or character? Seeing the change between them. And so this will come back, or this will be a prevalent theme throughout, I guess I would say. So first of all, some characters, obviously, characters, of course, being traits, DNA sequences, specific letters on the DNA sequence, the presence and absence of certain features, things like that, just traits in general. So some traits or characters are more helpful than others in your phylogenetic analysis. And of course, what these changes look like or how you model them depends on the assumptions you're using for that particular molecular or morphological model. And of course, because we're human and because nothing we do is perfect, these models of molecular morphological evolution are not going to be all encompassing. They're not going to be all applicable. Some are going to be more useful for some situations over others, some types of data over others. So that's some stuff to keep in mind. But essentially, these incongruencies, they I guess could be conflicting with the accepted phylogeny, but generally they're just noise in the data. So it's a situation where some characters are not supporting one relationship over the other, at least in terms of these specific discordant characters we're looking at. So just keeping that in mind as we go through this. Okay, so basically, and I'm going to need you to let me know if anyone has any questions or comments. Scott, if you can handle that, please.
I will do that.
All right. All right. So these are the, I guess, 4 broad categories of the biological causes for phylogenetic incongruence, and this is by no means exhaustive. You could go into much more detail about this, but I wanted to go into a basic layout of the causes here. So the first one kind of involves general, probably the one we've heard about the most, if any, convergence. So for instance, convergent evolution, when 2 pretty distantly related species evolve the same character state or trait, when their most recent common ancestor didn't have that trait, so it evolved independently, basically. And of course, this is often, but not always, due to similar environmental pressures and mechanical demands of the specific habitats they're living in. So living in water requires certain overall morphology, certain traits in order to survive and reproduce, basically. So basically, this means that those characters that are easier, quote unquote, or more frequently evolve more than once aren't always helpful. Because again, if you go back up here, what I said earlier about comparing unique evolutionary changes, basically, there are specifically shared unique traits, also called synapomorphies. So, for example, if you have one lineage, that has one trait, and it's only found that lineage compared to others, that's not particularly useful for telling the relationships between those different lineages because it's only found in one thing. Likewise, if you have one trait found among all the lineages, that's also not particularly useful because, well, it's shared by everything. That doesn't really tell you what's more closely related to what. So it's these shared, unique traits that are shared by some, but not all of the lineages in whatever study we're doing that are most useful here.
So that's the difference between synapomorphy and symplesiomorphy?
Yes,
basically?
correct. Yes.
So symplesiomorphy would be something where they all had that trait.
Right. Yeah, it's relative, of course, because it depends on what species you're looking at in your analysis. But yeah, if all the species in your analysis have that trait, you can consider it a symplesiomorphy because they all share that, likely because it's the ancestral state. They all inherit it from a common ancestor.
I need to interject that I think you're making the word symplesiomorphy up. I don't believe it. It sounds like a made-up word.
I did make it up just now.
No, it is an actual term in phylogenetics.
Okay, since you say so, Andrew, I will believe you.
All right. Well, if you don't believe me, look it up.
No, I trust you. I trust you.
Those two terms are the most confusing terms, I think, in phylogeny and in biology in general, basically.
Yeah, sometimes they can be.
Because the
basically, to reiterate, basically, you have apomorphies so you can consider, for example, the plesiomorphies, the ancestral state. So that's what everything in your analysis once had, or what the common ancestor had. Now, if you're considering it branching into the future, basically, one lineage will inherit one mutation or change, for example, and none of the other lineages will inherit that change, of course, with exceptions, as we're about to talk about. And that change will be unique to that lineage. So it's a derived state, an apomorphy. Now, if that continues, if that lineage with that change then branches again, then those next two lineages will also share that change. Hence, it's a synapomorphy, a shared derived trait. If that makes sense. I hope it does. So basically, as an example of convergent evolution, we can look at fur color, body shape, tooth design, where they're flat or cusped. These are the general characteristics that we think about when we think of animals, essentially, at least their superficial morphology. Of course, the creationist conception that, if you just compare how big something is, what color it is, what overall shape it is, that's not going to tell you anything. Well, of course it's not going to tell you anything, because these characters evolve or can change back and forth really quickly, or easily, I should say, because they're not that complex, relatively speaking. So, for example, you can look at this skull here and this skull here, and they look pretty similar. The teeth have these serrations and cusps needed for a carnivorous lifestyle. But of course, this is the skull of the marsupial wolf, basically, the Tasmanian tiger. And this is, of course, a wolf skull, placental mammal. So they independently evolved this, even though they look completely, pretty similar to each other. Now, of course, what's the solution to this? Include more than one character in your analysis. For example, another common example is if you were to just look at the trait of feathers or wings, or wings more specifically, you could group birds and bats together because they both have wings. But of course, that's not really accurate, because many other traits of birds indicate that they're more distantly related to bats than bats are to, say, other mammals, for example. So just comparing one trait to another isn't always that useful, because they could have independently evolved. By comparing a large data set of character states, basically doing a lot of math, that you can say, okay, well, they independently evolved this trait, but the other characters in the analysis say that they're not that closely related. So that's convergence basically there. Another type of convergence is character reversal, where we were talking about derived and ancestral states. Basically, a derived state goes back to the ancestral state. And this is particularly common for data sets with a limited number of character states. For example, you have DNA sequences with only 4 character states, A, C, T, and G. And if you have species that branched off a long, long time ago, and well, we'll talk about this more later, but like if you only have 4 character states of those 4 nucleotides, then they can go back and forth. They can mutate between each other quite readily. So that's another type of convergence there. Here's an example of this where like this is the ancestral sequence led to this species, and these are the this is the derived sequence, for example. But this one, after a long time from the node, the branch point, evolved a return to the ancestral state. So if we were just comparing those 2 sequences, we'd say, oh, this one is analogous to this one. They share a common ancestor. But of course, we don't do that because we compare multiple traits at once. So that's why doing a large analysis is handy, to say the least. So the next type of general type of incongruence is crossing between species where genetic material is shared. And probably the prime example of this is horizontal gene transfer, where you have usually 2 bacteria or archaea, for example, most common in those types of species. 2 individuals or even 2 distantly related species of these single cells can exchange genetic material so that, again, as before, like the reversals, they appear related to each other, but they're actually distantly related to each other. So to kind of put it loosely again, the uniqueness of these sequences has been erased over time because they now share something that their common ancestor didn't share. And you can find this in some plants and animals, but generally more common for prokaryotes. There's a picture of that, basically. Vertical gene transfer and horizontal gene transfer. If anyone says that I should go back to an image or something like that, please let me know, Scott. And of course...
Will do. I'm copying a link right now, so I may have missed a couple. But it looks like we're caught up with comments.
Okay, just making sure. Of course, you have hybridization between different species with the same problems as lateral gene transfer between plants and animals, for example, lions and tigers, and bears, like polar and grizzly bears, for example.
I think the problem with all of this is that with the long history of life, a lot of information can be erased, and that
we're about to get to that.
Okay, good. Problematic.
Yeah, but that's a good point there. But yeah, basically, just get into that with the next thing. We have what's called long branch attraction, or just long branch effects, basically. So if you have 2 species, for example, if you have an insect and an octopus or a vertebrate, for example, you compare those 3 species, they branched off from each other a really long time ago, like hundreds of millions of years ago at the very least. That's when they shared most recent common ancestor. So since then, substitution mutations have had long time to occur in single base pair or just sequence in general. And like we were saying earlier, there are a lot of reversals that in the DNA sequences, reversals can be pretty common because you can have, say, come back up here. Back to the other image. Here, yeah. So again, you can have lots of changes here, and you can have these reversals over significantly long periods of time, but more significantly, here, you can have, say, these two branches, and because there's been a significantly long time since they've diverged from each other, they have accumulated enough changes that overall it looks like they have similar changes, basically. But these, of course, are not similar through descent. They're similar because of chance mutations, basically. Because if you have enough time and you have a limited set of units, basically, there, then eventually you are going to get some convergences just by sheer probability. And of course, when you put this into the analysis, and okay, what is the most parsimonious tree, the tree that has the fewest number of character conflicts, of course, these are going to be branched together, when in fact, this is the true tree, and this is the tree that we get. Because of long branch attraction, we get incongruencies here. And this is also termed saturation, because the sequence has been saturated with mutations, and they look more closely related than they actually are, because the sequences are more similar than they were a long time ago. So a potential solution to this is to use slowly evolving genes or characters in your analysis. So if you have genes or sequences or molecules like proteins that change or accumulate changes at a slower rate, basically, then of course they're going to have less of these long branch effects, because they're yeah. And so the final type here is basically small scale effects, the main one being incomplete lineage sorting, where basically you have a common ancestor, and there are lots of versions of a gene or different alleles in this population, basically represented by this gray bar here. So that's the population lineage. Basically, you have multiple states of this gene, and if they speciate, sometimes just by chance they can become expressed in the diverging lineages, or as they're speciating, basically some can be found in this lineage, some can be lost in this other lineage. Basically, they can be what's the right word for it? Basically, help me out here.
Maybe there is no word for it.
Yeah, maybe there is no word for it.
Yeah, they ran out of words just before they came up with that concept, so there's no more words.
Right. Yeah. Basically, and you again get convergence there, and basically because of the speciation process, and because of random segregation of different alleles, because genetics is pretty stochastic, pretty messy. You can have gene trees, the basically the family trees of individual gene sequences that conflict with other gene trees from other gene sequences, and therefore conflict with the main species tree.
Yep.
And this can be amplified by rapid speciation, essentially. If you have lots of species radiating outwards in a geologically short time interval, basically. And this is most common for closely related species, but can also be found, like, for example, when you go back to the split of placental mammals, it's generally thought that the reason why it's so difficult to say, okay, the group that includes elephants and their relatives, the group that includes most things we think of when we think of placental mammals, like dogs and deer and humans, and then the other group that includes sloths and anteaters, for example, Xenarthra. The general reason why the root of that placental mammal phylogeny is so difficult to trace out is probably because of incomplete lineage sorting. Because you go back, dinosaurs went extinct, or mammals were radiating outward in this time, and that kind of caused incomplete lineage sorting, and so it's very difficult to figure out how these large mammal groups are related to each other, what the branching pattern specifically is. That's not to say that we don't know that elephants are more closely related to manatees, basically, than they are to like tapirs, for example, or just any other mammal group you can think of. So the broad relationships of each individual group are pretty resolved, but the overall mammal phylogeny is still being worked out, I guess I would say. That's pretty much it. And if I stop sharing there.
Is that a document that you have?
Oh, yeah, I just made it right now. It was a PDF.
Yeah, if you could send that to me, that would be cool.
Oh, okay, sure.
He wants to put it on his side of the refrigerator that's dedicated to you.
Yes.
Okay. Well, what in particular with like incomplete lineage sorting, for example, or just phylogenetic incongruencies in general, it's actually interesting because when you dig into it deeper, it's actually more a problem for creationists than it is for common ancestry, or the idea of common ancestry, I should say. Because generally, young Earth creationists, of course, we all know, believe in kinds of giant, or that's not the right word, family order level relationships, but anything above that doesn't exist, basically. Yeah. Well, if we were to take their argument to its natural conclusion and say, okay, these broad level relationships of, say, mammals, birds, reptiles, things like that, if incongruencies at that level mean that they aren't related, if there's no support for those relationships at that level, then we could just as easily say there are also minor incongruencies at the family, the species, the genus level, and therefore kinds, the members of those kinds aren't related to each other because there are incongruencies there. So pretty much the argument for incongruencies isn't that useful because, well, this kind of gets into the analogy I want to get over. I've been talking a while, so I'll shut up in a sec. But basically, the analogy I want everyone to consider is that, say you have free rocks, okay, just plain ordinary rocks, and presumably they have little deviations and little holes and gouges in them, just like all rocks do, okay? Now you're going to take your a small magnifying glass and look at them and say, okay, this one shares more in common with this one, or it looks similar to this one. Say, for example, pretty much the magnifying glass lens there can be considered our data. Basically, that's helping us resolve these relationships. And of course, the individual rocks are the phylogenies, essentially. Now, if you look with a really small magnifying glass and look at those rocks, you're gonna see broad categories there. and similarities and differences, for example, but you're not going to see the minutia of it. However, if you use more data, for example, if you use a bigger magnifying lens or even a microscope or things of that nature, you're going to see those holes, those gouges, and say, okay, this one's different from that. In this case, this phylogeny is different from that. This is the more correct phylogeny. Of course, if you really want to take it up a notch, instead of considering free rocks, you can consider 10 million, 100 million, a trillion, 10 to the 80th power when you're getting to that many taxa of rocks, or in this case, of phylogenies. So basically, long story short, phylogenetic incongruencies, not a problem for common ancestry. And I hope, at least in some small way, that if anyone, the audience, or just anyone in general listening to this, is ever confronted with that type of argument again, they can at least have a better idea of where to come at it and confront it and say, okay, no, this is just wrong. So yeah, that's pretty much what I want to say.
Yeah. So the common charge, and I hate to utter this phrase, is rescue device.
Right.
That incomplete lineage sorting, gene conversion, horizontal gene transfer, all of that is just rescue devices. But what they actually are is the more knowledge we have about all of the different things that can happen historically, the more we understand how these incongruencies can develop. And if that's a rescue device, so be it. It's a rescue device that is based on knowing things, knowing how things happen.
Another good example of that probably is a simpler one. Like if you have parents with one eye color and they give rise to a offspring with a different eye color, for example, that could be considered incongruencies in the gene trees, for example, or just small scale stuff like that. But yeah, like you said, like if we're taking those seriously, these aren't rescue devices because we observe them in individuals, in populations today. Like, oh yeah, co-option too.
Yeah, there's an amusing in a Yale University course on evolution that's not for biology. I think it's for medical people. Stephen Stearns does an amusing thing about eye color where, his wife has brown eyes, he has brown eyes, and all of the males that she knows have blue eyes, and his child has blue eyes. Should he worry? And he goes ahead and explains why he shouldn't worry, why that can happen. Basically,
right?
It's
exactly.
Kind of a real interesting example. But.
Right. Yeah. Basically, the stochasticity of genetics and of the gene trees that correspond to them, we don't expect them to always line up with species trees just because, well, nature is messy. And I mean, like, if you're going to say basically that, oh, well, our methods are imperfect, they're not 100% accurate all the time. Well, that's not what common ancestry predicts. Basically, like the more specifically, what you're trying to find, mentioning those millions of different potential trees, what you're trying to find is a statistically high phylogenetic signal. And well, that term I've mentioned before, but like that's the key thing here. It's all about statistics. And if you're going to point out some incongruencies and variations at, well, the small regions of the tree of life, then you're really not doing the subject justice.
Yeah.
To be honest.
And then thinking about geology and rocks, I think about rocks a lot. Like every rock you pick up is just like totally unique, right? And has its own. But I mean, if you imagine, okay, a mountain range rises up because of two plates colliding, and then over a million years it erodes, falls down into a lower area, forms sediment, and then that sediment basically gets buried by kilometers of other sediment, and then it changes its morphology based on the weight and the temperature, right? And becomes a metamorphic rock. And then something else rises up and basically sticks that back above the ground, and it wears down, so that now you have a wear plane, and then it cracks, and then a glacier picks it up and moves it into Minnesota. And then I pick it up and look at it and like, what the fuck?
Right.
What is this thing's history? Yet geologists are able to piece together a great deal of that history by looking at the macro structures and looking at the history of all of that, and looking chemically at what's in the rock. And they can figure out, we have a syncline, anticline, all of these amazing things that you can figure out when you pick up a rock. It's kind of cool. Yeah.
Right. That's a good point, because, like John Maddox likes to say, oh, well, you're not looking at the macro view, basically. Well, it doesn't really help to look at the macro view, and it doesn't really help to look at just the micro view. You kind of have to consider the whole thing. Yeah, exactly.
Yep. Yeah, and imagine if rocks duplicated, if they had sex and combined, right? And formed other little rocks that were like them, and we had populations of rocks moving around the earth, that sort of thing. That would be life, So life and geology have a hell of a lot in common as far as the randomness or stochasticity or the historicity of what happens, and it's interesting to think about. Be cool if rocks did reproduce, wouldn't it? Like if they got together and, had a little romance and I don't know.
Tell you what, stupid pardon turkey energy just popped in, has just been throwing the knowledge at us ever since.
There's an ERV. Yeah, that's the one that that SFT likes to throw in, the gorilla and chimpanzee. There's one ERV, I think, that was because of incomplete lineage sorting.
Yeah, I was looking at one of SFT's videos now, where we're mentioning him, and he was saying something along the lines of, "Oh, we're finding lots of character loss," and he mentioned what you were talking about earlier about rescue devices with convergent evolution, for example. That's what they always go to. I mean, like. And he was specifically saying, like, this was one of his older videos, but he was specifically stating that the more we look at molecular and morphological data, the more incongruencies we are finding, which isn't at all the case. But
it's the opposite.
Right. But like, here's the thing, like, what bothers me the most is that they're saying that the nested hierarchical pattern that we see in life is neutral to the debate, quote unquote. But like, if a nested hierarchy is just as good evidence for design, whatever model of design you're using, as it is for common descent, according to them, then why would we expect these incongruencies in living things as well? I mean, like, make up your mind here, because like, what's evidence for your model? Incongruent pattern in life or a congruent pattern in life? You can't have it both ways, or at least that's my confusion here. Like, I don't know. Am I making sense here?
Yeah. What I would expect if creation was true, and if God created every species, and he sat down and decided to just start creating a batch of animals, I would expect all of them to be vastly different from one another. I wouldn't expect him to use the same things over and over again.
Right.
Why would he have to? But if you're to accept their idea, you have to believe that God woke up one day and he decided, oh, today I'm going to create dog-like things, right? So I'm going to create a bear and a raccoon, and I'm going to, create things with floppy ears, and and then the next day he decided, oh, I'm going to create elephant-like things, like I'll create a rhinoceros and an elephant and things without a lot of fur, and like, I mean, none of that's in the Bible about God waking up one day and thinking, oh, I'm going to create this kind of thing, but not just a kind. I'm going to create a number of kinds that kind of have a lot to do with each other, right? They're very similar.
Right.
And then tomorrow I'm going to erase all that and start creating the giraffe kind, different kind.
Right. And modifying traits in ways that would exactly corroborate evolution. Like it's
right.
If a common designer does indeed exist, I'm just going to say he is the most unoriginal, uncreative designer I have ever seen. Right. Because if he's really omnipotent, there are a million different ways he could have designed the patterns we see in life. And surprise, surprise, the one we see just happens to be almost the exact pattern that we would expect of common ancestry. Now, why is that?
Yeah, life could have been so much more interesting if we had many different kinds of things.
Okay, stupid pardoned turkey energy just threw in some controversy here. It's heating up here at Answers in Atheism.
Oh no.
I'm pretty sure Aquatic Apes was not supported, wasn't it? I'm not a primate expert, but like,
no, I don't think it has been. I think it's pretty unsupported.
She's just a troublemaker.
She's a troublemaker. Yep. Kind of like Germania. He's a troublemaker.
I don't know you're going to bring it to that.
Yeah, that asshole never came back. He said, I'm going to go to the car and clean it out. I guess he got sick off the French fries he found on the floorboard.
He probably got sick from listening to his stream last night. I know I did. I have this habit of, like, I leave a stream up on the TV or on my monitors, right? And I go watch TV, and every now and then I'll hit the unmute button to hear what they're talking about. And last night I hit the unmute button and I wanted to pull my ears out and scrape my eardrums out of my skull. I'm like, why? I mean, either the entire stream was like that or I just had really bad timing. I don't know. Anyway. Yeah.
So should we open it up, seeing if anyone wants to join, and then if not,
I sent an invite to Nestle, but he didn't respond, and we could open it up or kill it.
Yeah, I'll put it out there and see who wants to come in and discuss science stuff.
Sciencey stuff. So, Topoi Samare's favorite. That's his favorite, right?
Yeah.
Well, it turns out
it's in the Gospels, apparently, somewhere there around the lilies of the field or whatever. It's out around there somewhere. It's in the Gospels. I didn't know that. I'm just not that sharp of a hermeneuticist to draw that out.
Are you making a joke, or did he actually say that?
Well, he made topo. Say it again for me, Speed.
Topo isomerase.
Topo isomerase sound like this spiritual thing. It was just weird. It's just weird when he talks about it, and then starts doing lilies of the valley or field or whatever.
If you look into the evolution of our idea of topoisomerase, right? I think it was discovered in like the 50s or 60s, and then zinc fingers, which have a lot to do with topoisomerase. Anyway, topoisomerase is as old as DNA replication, I believe, and so it's been around for a while. So we find different families in Prokarya, Archaea. Some people say Archaea, and basically eukaryotes. And there are, I think they've discovered so far 6 or 7 different families, and they've looked at they've got phylogenetic trees for the evolution of topoisomerases, and it appears that it developed more than once. They think there's 2 different ways for it to develop. And I was looking into the evolution of topoisomerase, which apparently Cell has never done because he insists that it can't evolve.
Right.
It had to be a certain way, and this is not the case at all. When you look into zinc fingers, zinc fingers are the most common motif of all protein motifs that there are. Basically, it's basically 2 histidines and 2 cysteines that exist somewhere on the chain, separated by enough that the DNA can, or the protein can fold back on itself and grab hold of a zinc or an iron or some other metallic ion, right?
Wow.
And what it does is it sticks it together so that it forms like a loop, and that loop can be like a knuckle that attaches to a piece of DNA or RNA or even a lipid, and has, I think there are 700 different zinc finger proteins in the human body. So when you start really looking into the evolution of this stuff, you find so many homologies and so much much phylogeny.
Right.
That
well, like they also mentioned like orphan or taxonomically restricted genes, for example, and you still see a nested hierarchy with those.
Right. Yeah, and orphan genes are something that, you know, they throw out there. Like I think they found like 13,000 orphan genes, and they throw it out there like, we found function for these orphan genes, and now we know that they're not orphans, and that they don't just, not do. Well, we found function for about 24 of them out of the thousands, right? And we know they're orphan genes because we know that they have lost the ability to express themselves in a lot of cases.
Just real quick. Correspondingly, like with pseudogenes, for example, you could easily say, "Oh, we found some with function," but the important thing there.
That's what I meant to talk about. I didn't mean.
Okay.
Though there's
like just non-functional regions of the genome that they turn out to have some minor functions, but like the important thing here, at least with relation to design versus common ancestry, is whether or not those functional elements or those functions specifically are correlated with the specific organisms. If they have if there's some kind of pattern there that we can notice, and of course we haven't seen one so far. So,
yeah, and there's all sorts of possibilities. Even a pseudogene, if it can express, if there's some way for it to express, it's going to have some effect on the cell.
Right, like it can acquire additional functions later on.
But it could have a function. Like if you think of functionality, and you think about the reason why junk DNA is such a bad topic, it's the same as, when did you quit beating your wife? It's like the functionality exists on a scale from 0 to 100. Something that is absolutely essential, and something that can completely be cast out, and somewhere in between lies the answer. So, I mean, functionality is a really deep topic that they like to make it binary. Either everything is functional or everything is not, that sort of thing. And it's nowhere near that simple. Very, very complicated. My favorite pseudogene is DDXL11 or DDXL2, I think it is, in the chromosome 2 fusion. And that particular pseudogene is like a little tiny sliver off of a very big DDX11 gene, which is a dead box. It has dead box motifs in it. And the pseudogene itself, even though it expresses at some level because it has promoters, it only expresses, I think, mostly in the testicles, where it isn't really used at all. The thing is, it has no structural components to it from the original gene that it could possibly use to be any kind of functional. So it probably has zero function, doesn't do anything at all. And that's the one that they point out as being right at that fusion site. Right. And it's just, and they still, SFT as recently as a month ago called it a highly functional gene. And nobody has ever found a function for this gene. None, zero, nada. It's just like, they love to lie.
Yeah, or like what you were saying earlier, like sometimes function matters, but sometimes it doesn't. What I think is at least somewhat more important is whether it's essential or not, whether it affects fitness, right?
Right. Well, yeah, and even that is, on a scale of 0 to 100 as to what's going to affect fitness. Because you could have a really highly beneficial, absolutely essential gene right next to something that's actually detrimental. Right. And those things, because of recombination, are always going to ride together. So you're always going to have this slight, wrench in the works, that's riding along with it.
Right.
But the organism lives, and fitness is all about whether or not the organism lives, and reproduces, that sort of thing.
Or Dessel here, just like Mendel's accountant is the primary pop gen simulator in the field.
Exactly right. Yeah, nobody uses it except these guys. It's horrible.
Hey, Peter.
Anyway. Hi, Peter.
Oops, I was muted.
You're Einstein.
Hi, Peter.
Yeah, my icon would indicate otherwise. I didn't see anything because I just noticed the show and then you put the link. But I will rewatch it, but I think that I'm far too stupid to get any of that thing.
Oh, no, not
that's okay.
It's still interesting. It's still interesting, and I will still watch it.
I would like you to make the document available, Andrew, and maybe I could find a way to link it to the public and put it in the description, because it's a pretty damn good document you did there. So.
Oh, really? I just put it together in like half an hour.
Well, there you go. You're a genius. I guess.
I wouldn't say that.
Well, no genius would.
If you want, I'll donate my icon to you.
Okay. Yeah. There you go. Well, we're close to shooting it in the head, Peter.
Okay. Well, so can I then have an additional topic in this show, which I think you could not do without
Andrew's permission? Yeah.
Or give me the a taste of it, and then we'll see if we want to do it.
Yeah, we'll decide.
Hawaii shirts.
What?
Hawaii shirts.
Hawaii shirts.
Hawaiian shirts.
Hawaiian shirts.
Oh,
yes.
Okay.
Don't talk shit about Hawaiian shirts now.
that should be a good clue.
Hawaiian shirts. Oh no,
Hoven.
Not him. Hoven.
Oh. I like Hawaiian shirts. I they look better on me than Hoven.
Well, not necessarily, but it is a clue. So, I am on Twitter and I'm on YouTube. Strictly speaking, I also have a Discord server and a Facebook page for my channel, all of which are completely dead. But so people have been talking to me about this new fangled thing that they've recently discovered, which is TikTok. And I don't need another platform.
Nope.
But now there's some guy who made it really, really difficult for me not to go on TikTok. And so, Speed, can you please make me full screen for a second? You will not be disappointed. I know you.
Okay.
Do I need to get a fork to poke my eyes out?
Okay, you promise I won't be shocked.
Well,
I'm getting a fork. When Peter comes on, I need a fork.
No, don't walk away. You need to watch this in full.
Okay, I've got a fork right here in my hands.
Yeah, everyone needs to watch this in full.
I always tremble when Peter's full screen.
Oh, I've seen this. Yeah.
Oh, the weather outside is frightful, but the fire is so delightful. And since we've no place to go, let it snow, let it snow, let it snow. And it doesn't show signs of stopping, and I brought me some corn for popping. Yeah, maybe it'll evolve. The lights are turned way down low. Let it snow, let it snow, let it snow. Okay, here to help.
Okay.
Oh man, that's so cheerful. It puts me in a mood.
Oh, that's great.
If anything will put you in the Christmas mood, this will.
I heard a rumor about that last night. Yeah, I hadn't seen it.
He needs a YouTube channel so everyone has access to stuff like this.
Yep.
This is just fucking great. You can take me off screen.
Okay.
If you want.
I wonder if any of Megan's family has called to reassure her that they'll take her in if she just gets a little more ashamed of having a guy who being married to a guy who does that kind of stuff. Because to be fair, if you go to his TikTok, he has a rap on there as well. And then he has the complaint video that someone's impersonating him. I think it's brilliant. I think we need a lot more of that. Yeah, it's very cool. So Crip, well, what's the word when a small group of people know what that's about? You know what it's all about? I don't know. Esoteric.
Esoteric. I don't know.
I don't know. Sorry.
Nick?
I don't know either, Scott, but I can tell you what the word is for a group of creationists.
What?
Well,
disappointment.
Oh.
Oh, excellent. Well, guys, there's a debate tonight with Dr. Dan and Snowflake or Snowflower or
Sunflower, I believe is his name.
Sunflower.
Like,
what's the topic?
Oh my God! Have you ever listened to Sunflower? The topic is going to be hilarity. I think it's
viruses and evolution.
By the way, speaking about debates, have you mentioned the debate that Erica did?
She did a debate last night, right? Was that last night?
Yeah, she debated last night, and as predicted, Nephilim Free ran from the debate. Mid-debate, he just left.
Okay.
Is that Central Time, Scott?
That's 8:00 Central Time. I'm sorry, I did not specify.
Yeah.
But I'm looking forward to that. I think I'm going to try to catch up on the Erica and that guy Jordan. Reasons to Doubt, or I think is his name, is his channel name, was also a co-debater with Erica. Teamed up.
Where was Erica's? What channel was Erica's debate on?
Modern Day Debate.
Modern Day Debate. I put the link to the Sunflower versus Dr. Dan. I'm so sorry for Dr. Dan.
That's already in the description. And
okay.
Yeah, we have to find the. But we're going to do an after show after the Sunflower debate. So.
Okay, I'll put the link. I'll put the link to Erica's debate in the chat as well. There you go.
I thought you already did. Which one is it?
The last one I posted.
Okay, got it. Good. I'm gonna go check that out too. I missed that last night. I was watching television. Anyway, so I think we should all take a break and shoot it in the head. What do you think?
What's television? Something new?
Television. Yeah. It's the old stuff. You get the rabbit ears out and you put the rabbit ears.
It's big YouTube.
You adjust the rabbit ears until you quit seeing the snow and you see the signal, and then you watch some good stuff.
And if you have to, you have your youngest child just stand next to the TV holding a copper wire that's wrapped around the antenna, and that might fix it.
Yeah,
it's a rectangular box that helps in hypnosis of certain individuals.
Yep.
Also known as a gaming console.
So,
that too.
We moved out to the country and we did not have an antenna on the roof back in the day, back in like 1979. And so I made one out of aluminum foil and I cut it to the exact bandwidth of the TV station. And it actually worked very well. But I had to have my children hold it up while I watched television. And they hate me to this day. They still complain about that.
But then why else have children? That's what I don't get. Why would you not? Why would you have children if they're not going to hold up the antenna for you?
That's right.
I'm more confused that Speed's part of the aluminum foil community.
I am.
Worrying.
Well, I have a tinfoil hat here. You know that, right?
Of course you do.
I do have a tinfoil, and I have aluminum foil underwear because I understand that females are aliens and they beam waves into your testicles and make you do stupid things. So I always wear aluminum foil underwear.
That sounds legit.
They're from Venus.
Definitely. Well, everybody else does it too. They're just too ashamed to admit it.
I know. Yeah. Yep. Anyway, so on that note, guys, we're going to end it.
Okay.
So goodbye.
Great discussion.
Thank
Thanks,
you for
Andrew, for putting all that together.
Yeah, thank you.
Yeah. I hope it was entertaining or educational for at least some people.
Unlike most things we do here, it was real content.
Just one small piece of criticism. I just went to the start of the stream.
What?
You used the wrong intro again.
That's fine. I used the regular show intro, right?
Yeah, but this is a special show, so you shouldn't use the regular show intro.
So I should use the. I got.
And it's Saturday. Yeah,
but that's a Snada intro. That's different.
We do need to come up with an intro just for.
So what you're saying is what Rational Empiricist just brought to your channel is not worth being put in the Saturday night.
Don't do that, Peter.
Well, no,
I think he should give his own intro. There should be a Rational Humanist.
Wait a minute.
Rant time intro.
Okay, let's just throw that intro on here. Okay, demonic irreverence. Did you see that? That's not what Andrew does. So I couldn't use that. No, he doesn't do demonic irreverence.
Well, speaking of that, how many people have noticed the Easter egg yet? Don't tell them what it is, just how many people have seen it. Speed.
What?
The Easter egg in that intro.
I know.
Is it an Easter egg?
I love it, actually. I wish you'd make it a little more obvious.
No. But how many people have seen it?
I have.
How many people have contacted you and said, Speed, do you know that that's there?
None of them do.
No.
I think they assume that I put it in there myself.
That could be. But it's the second one that I've ever made that has an Easter egg. Yeah. The first one has been online for 3 years now. No one has noticed.
Well, you'll see it tonight at the beginning of Snada, which is going to be the after show for the debate. So anyway, I'll try to do it right.
Saturday night at the after show.
There you go.
So Dessel, I assume you came in because you know we're ending. Okay, we got to end. Goodbye, cruel world. We're ending this thing.
The YouTube channel